It's a pleasure to be here to tell you all about a microscopy technique that we developed to help scientists take better images faster. So I want to start out by saying that microscopy is one of the most important tools in all of biology. The microscopy in life sciences market is growing at about $2 billion today, spanning academic labs as well as biotech and pharmaceutical companies. And what's more, the use of super resolution microscopy is growing at a commensurate rate. And the reason for this is really, like Rana said, that scientists are starting to more and more study systems on size scales that are too small to register in standard microscopy. And so here we're looking at a couple of cells stained for a structural protein. And we can see that with the application of super resolution microscopy, we can really start to pick apart those individual fibers, kind of as an example. So there are a number of super resolution microscopy techniques that exist today, but there are sort of two key limitations that are ubiquitous in the field. And the first is limited access to the multi-well plate formats that scientists really need in order to run large-scale experiments like screens. The second is that these techniques require highly specialized equipment as well as protocols and materials that researchers are typically unfamiliar with. Now there is one super resolution microscopy that was developed in 2015 in Ed Boyden's lab that obviates the need for a specialized microscope. And that's called expansion microscopy. And to explain how it works in an analogy, imagine I have a balloon and I write a word on the surface of that balloon and then inflate it. Well, as that balloon inflates, the word sort of increases in size and we get a bigger word that's easier to read. Well, we can do something similar in cells by embedding them in a swellable hydrogel. And as that hydrogel swells, so too do the cells embedded within, and we get cells that are essentially larger that we can effectively image at a higher resolution by virtue of that fact. We can see an example of this happening here on a slice of mouse brain tissue. Now this is a fantastic technology because it allows researchers to do super resolution microscopy using the same protocols and materials and even microscopes that they're already using and already comfortable with. But throughput has still been limited to the preparation of individual samples on glass slides. So we took it upon ourselves to develop a technique that puts expansion microscopy directly in the wells of a 96 well plate to solve the throughput issue in super resolution microscopy. And the device that we came up with, oh, there's the timer, is this plastic well plate insert. We have a patent filed for it. The fact that it's so simple makes it super inexpensive to manufacture. And it works by collecting small volumes of this gel solution in a manner that allows for, and depositing them into the wells of the well plate where the cells are cultured, in a manner that allows for the expansion process to happen without any manual handling of these fragile gels. And that really allows us to produce hundreds of these gels across well plates in the same time frame that it would normally take us to produce just one. So we think that this technique has the potential to be massively beneficial across the field of cell biology. But to give you one example of something that we're particularly excited about, we see an immediate need for this technology in the drug development pipeline, and particularly in preclinical development where researchers are doing experiments on cells in dishes. And to be a little bit more specific, one of the most important types of drugs that are being developed today are these nanocarriers, which serve as vehicles for therapeutic payloads. But these vehicles have to get inside of cells through an internalization process, after which they're trafficked through convoluted highways of vesicles. And this trafficking process often governs the effectiveness of these drug vehicles in ways that are poorly understood. And when we take a look at how this process is typically studied, we can see why this process is poorly understood. So what you're looking at is a couple of cells that were imaged before and after the application of our device on the top and bottom respectively. And we're looking at a couple of the building blocks of this highway that I'm talking about inside cells. And what I hope you can appreciate from these images is that we really need, the super resolution in order to pick apart this highway in a manner that would give us useful information about the drugs that it carries. So this is just one application for our device that we think could be impactful. If you're interested in hearing more, please come visit me at our poster We're looking for connections to potential end users, be them in academia or biotech or pharmaceutical companies. We're also looking for connections to potential industry collaborators who would be interested in taking this, as of yet, simple device and moving it into more of a black box automated platform. And thank you for your time.